reads, "Explain like I'm 5." So here is the long-awaited explanation regarding soluble ACE2. So while I'm desperately trying not to take that completely literally, there is some background context I need to give you. So call me Miss Frizzle and let's hop on this learning express. We are made up of cells. This is one of our typical cells. These parts I'm sure are familiar, like the nucleus that holds our genetic material or our DNA, mitochondria, which are the powerhouse of the cell that make energy. Now we're going to look at the cell membrane. Each cell is like its own manufacturing plant or warehouse. So to keep that warehouse in order, we're going to need a structure around it. The cell membrane is made up of a phospholipid bilayer, meaning it is made up of bilayer, 2 layers of phosphorus, and lipids, fats. Each of these phospholipids is made up of a hydrophilic head and a hydrophobic tail. Hydro means water, while philic means loving, and phobic means fear of. So water-loving heads and water-fearful tails. So the water-fearful tails clump together, and the water-loving heads anchor it. In this form, it makes it incredibly difficult to import materials that the warehouse would need, and increasingly difficult to export materials that the warehouse makes. In order to adjust for that, we have something called membrane proteins. And the membrane protein that is important to this discussion is the receptor. The receptor binds to chemical messengers, such as hormones that are sent by other cells. Often these messengers need to get within the cell so the cell can act. So the receptor grants entry. Specifically, one of the receptors that we have on our cell membranes is called ACE2. It's usually responsible for helping maintain our blood pressure. Unfortunately, the spike protein of the virus, which contains a receptor binding domain, can also attach to ACE2. In endocytosis or the endosomal entry, once the receptor binding domain binds the receptor, ACE2 pulls into the cell, engulfing it into a vesicle. Now that vesicle, which is made up of the cell membrane sac, essentially, gets broken down and the viral contents are unleashed. Viral RNA empties into the cell and begins replicating. Another way it can enter is through membrane fusion. With membrane fusion, the assistance of TMPRSS2 is required. ACE2 pulls down, begins fusing with the membrane, and TMPRSS2 that acts as a cleavage site, allowing the viral contents to be emptied into the cell. And cleavage means what you might expect, something that divides or separates, or pulls away or pinches apart. Now that we have truly important foundational knowledge, let's jump in. So the name of this paper is Soluble ACE2 Correlates with Severe COVID-19 and Can Impair Antibody Responses. We already know now that ACE2 is a type 1 transmembrane protein, and it can exist in either a cellular membrane-bound or circulating soluble forms. The article goes on to say that the disintegrin and metalloprotein 17, aka Atom17, cleaves the ACE2 ectodomain of the membrane-bound receptor, shedding it into the extracellular space. Atom is a protease enzyme that can do similar functions like TMPRSS2, where it divides, separates, or cuts apart. Now I have Atom17 here pictured in more of its representative form rather than its true form. While TMPRSS2 would cleave to make ACE2 and the viral contents enter the cell, Atom17 cleaves to make ACE2 fall off the cell and into the bloodstream. So that's what that scary sentence means. Atom17 cleaves the ACE2 ectodomain of the membrane-bound receptor, shedding into the extracellular space. Ectodomain is just out of the membrane, essentially. So the ACE2 that's on the outside, the Atom17 cuts off. Now, interestingly, the SARS-CoV-1 virus, the original SARS virus, in vitro found that binding the viral spike increased Atom17 mediated release of soluble ACE2. So with SARS-1, anytime the ACE2 receptor bound with the spike protein, Atom17 was more likely to cut off more ACE2. With SARS-CoV-2, we know that cleaved ACE2 becomes soluble ACE2 in the bloodstream. With the word soluble, you know that the concentration value of something goes down with more liquid. So dilute or disintegrate, for lack of a better word. Now, the problem with the soluble ACE2, once it enters the bloodstream, if there are SARS-CoV-2 viruses around, these soluble ACE2 fragments can attach to or bind to the spike protein receptor binding domain. Why might that be a problem? As I've mentioned before, the soluble ACE2 is able to taxi the SARS-CoV-2 virus all over the body. It's one thing to get transported far away. How is it not getting caught? It's a fugitive, remember? That receptor binding domain of the spike protein that attaches to soluble ACE2 is the same receptor binding domain that the antibodies target. So if the soluble ACE2 is already bound to the receptor binding domain of the spike protein, the receptor binding domain is now cloaked and the antibodies can't attach. And the antibodies cannot mount an attack to something they can't see. Now, if that weren't bad enough, remember that the soluble ACE2 that have essentially provided an invisibility cloak for the virus smuggle the entire virus away from the initial site of infection, where much of our immune response is occurring and many of our immune cells are surveilling. What happens to solubles in liquid over time? They dissolve. And now that the receptor binding domain is open, the virus is free to infect a cell that's much further away from the initial site of infection. The other thing this article discussed is not only do the antibodies have difficulty in targeting it, it may impact our ability to form antibodies to begin with. But I think I'm going to save that for part 2. Anyway, I hope that helped.